So our next one is, the speaker is Professor GuoJun Zhang from the Hubei University of Chinese Medicine, Wuhan. So the title of his topic is Reduced Graphene Oxide-Based Field Effect Transistor and Biosensor for sensitive detection of Ebola virus. Professor Zhang?
Yes. Thank you so much, Dr. Wei. Good morning, everybody. So I'm GuoJun Zhang from Hubei University of Chinese Medicine.
GuoJun, would you please put your slide on the presentation mode?
Okay.
Yeah, thanks.
I'm very happy. So I would like to thank Professor Wei to organize this workshop. Also, thank you for giving me the chance to present here. So today I like to present to you a reduced graphene oxide-based field effect transistor biosensor, which can be used for sensitive detection of Ebola virus and possible detection of COVID-19. So today, presentation outline is shown here. First of all, I like to talk about a little bit about background on the COVID-19. Then I will introduce graphene-based FET biosensor. Afterwards, I will present to you this biosensor can be used for detection of Ebola virus. Subsequently, I will talk about our ongoing project on detection of COVID-19. Finally, I will make a summary. Since the emergence of coronavirus late last year, it has outbroken in every continent of the world. As of the day before yesterday, you see the number, about 13 million people worldwide have been diagnosed with this virus. And about 570,000 people died from it. This means the global situation with COVID-19 is pretty serious. So question is, what is coronavirus? How does this virus infect human body? So SARS coronavirus 2 belongs to the beta coronavirus category. The average diameter is about 125 nanometers. This virus has a clone appearance due to the presence of the spike glycoprotein on the envelope. As you can see from the left figure, the main structure of the virus includes the spike, envelope, membrane, and nuclear capsid. Among the structure elements, the S protein plays a key role in the cause of infection. This virus enters a host cell by binding to the SAE2 on the cell surface via S protein. With 10 to 20-fold high affinity than SARS. Maybe this is one of the reasons why this virus is more contagious than SARS. In terms of the viral genome, this virus is a positive sense. This is actually a single-stranded RNA virus, which contains approximately 30,000 nucleotides. So among them, the open reading frame 1A and 1B encoded non-structured protein, while other sequence mainly encoded structural protein. It should be noted that 3 regions have been identified for the detection of coronavirus, which are the RD, RP, and the second one is E gene. The third one is N gene. So as is known to everybody, so coronavirus can induce severe pneumonia. For example, fever, dry cough, and chest pain are the most typical symptoms. But actually, not only can this virus invade lung tissue, but also the heart, kidney, intestine, and other organs. So in summary, COVID-19 may cause multi-organ failure in extremely severe cases. Therefore, in order to prevent the spread of this disease, early screening and diagnosis are particularly important for the treatment and the control of the disease. With the development of related research, there are a lot of detection methods already developed for this new virus. As you see from this slide, so the methods include immunologic detection, molecular biological detection, etiology detection, and image detection like CT. So we think recently in clinical laboratories, the combined detection is the main testing strategy. Now let's focus on the comparison of the nucleic acid and antibody detection. This table shows performance of nucleic acid testing and antibody detection. And you can see both of the detection methods have advantage and disadvantage. The main disadvantage of these two detection methods are high false negative red and high false positive red. Therefore, a rapid, accurate, sensitive portable detection device is highly required. Recently, exactly this year, a lot of new technology like nanotechnology and the biosensor are attracting the attention of many researchers. Many recent papers have been published on their potential application in COVID-19 diagnosis and treatment. Now, I'd like to introduce you 4 studies regarding biosensor for coronavirus virus detection. So the first one is a dual functional plasmonic biosensor combining the plasmonic photothermal effect and the localized surface plasma resonance. The gold nano islands functionalized with completed DNA receptors can perform a sensitive detection of RD-RP sequence, which I mentioned earlier. So the next one establish a portable microfluidic immunoassay system for multiple and on-site detection IgG and IgM and antigen of coronavirus simultaneously. So this integrated method was successfully applied for detecting 26 patients with COVID-19 infection. So the third example report colorimetric assay based on golden nanoparticle. Those probe kept a golden nanoparticle aluminate only in the presence of his target RNA sequence. Thus lead to a change in its surface plasma resonance. This biosensor can simultaneously target 2 of the engine regions. The last one reports a rapid and sensitive lateral flow immune assay, which use a lens-lined Doppler polycystin nanoparticle to detect coronavirus IgG in human serum. So very recently, a group from Korea reported a graphene-based FET biosensing device which can be used for detecting S protein on the surface of coronavirus. So the structure is shown here. After graphene FET is fabricated, coronavirus can be conjugated onto the graphene sheet via antibody and antigen interaction. And then the virus can directly detect it. This means FET biosensor would be a good platform for coronavirus detection. So what is the principle of graphene-based FET biosensor? So shown left is a category of many nanomaterial. So as you can see, so graphene is one of them, and we think graphene has a lot of advantages. For example, it has a large surface volume ratio. The mobility is high, and the biocompatibility is also high. And the right figure is a schematic diagram of FET. And this FET is typical 3 electrodes. And you can source, drain, and the gate. In between source and drain, so graphene can be connected. To detect the protein, specific antibody can be immobilized on the graphene sheet. As you know, antibody can be negative charged and positive charged. And this change, contribution of antibody to the graphene sheet can make the current change. And this is a typical empire polar curve. Okay. So before and after antibody antigen interaction. So the gate voltage can shift. So this means the information of antibody and antigen interaction can be changed to electrical signal. So this is actually the principle of the graphene-based FET biosensor. And the advantage of this biosensor FET, for example, the detection can be level-free, and the sensitivity is high. Specificity is also high. Very importantly, as this FET, because it is made on the semiconductor material. So this device can be miniaturized and integrated for portable device purpose. So before to make the biosensor, so first step is to make the FET chip. Okay, this is actually optical image of the 4-inch silicon wafer. So one or actually FET chip is fabricated on the this wafer. This wafer can make 110 single FET chip. This chip is quite small, and the figure C actually is the SEM image. Okay, this shows the sensing area. So one single FET chip has 6 individual sensors. Okay, this is actually sensor array, and the white line shows the electrode, and the sensing area actually is between the 2 electrodes, and the width of the sensing area is 4 micrometers. Before biosensing, okay, graphene is drop casted, you know, in between the source and the drain. And this is the first example. We use the graphene FET biosensor for detection of DNA. So in this work, we employ PNA as a probe. PNA actually is a DNA analog. Okay, as we know, DNA is negative charge, and the PNA doesn't have charge; it's neutral. This means if we use a PNA as a probe, so after PNA DNA hybridization, so the background signal can be minimized. This can increase the sensitivity, and this is a schematic illustration of the graphene FET biosensor for detection of DNA, and this chip, FET chip, and reduced graphene oxide can be dropcasted on the silicon oxide, and the PNA is immobilized on the graphene surface, and ethanamine used to block the unreacted sides of the nanomaterial graphene sheet. Then DNA can be hybridized with the specific PNA probe, and then DNA can be detected by monitoring the direct voltage change. So after dropcasting reduce the graphene oxide on the chip surface, so several characterization tools are used. For example, Raman structure and SEM, AFM, and Raman structure. We can see two typical picks. One is a D pick, and the other is a G pick. So SEM image show here, we can see very clearly the graphene nanosheet spin across the two electrodes. AFM can tell us the information of how high the graphene is on the surface. So from this AFM experiment, we can see. So the height of graphene sheet is between 1.5 to 6 nanometer. So this means the graphene sheet dropcasted on the chip actually is not a single layer; it's a multiple layer. To investigate the sensitivity, so we applied a different concentration from 1 nano to 10 femtomolar to the PNA immobilized graphene FET biosensor. And after the hybridization, because the DNA is a negative charge, and actually this is a contribution of p-doping, and the direct point shifted to left. And along with the concentration of targeted DNA, and the signal also increased. And from this results, we got the LOD results, and we confirm 100 femtomolar DNA was detected. Also, we investigated the specificity. Okay, so in this work, we choose different targets. For example, non-complementary sequence, one-base mismatched sequence, and a complementary sequence. So from this results, you can see the signal from one base non-complement and the control smaller, much smaller than the signal of complementary sequence. So this specificity is also good. So the second example I show you here is we use this graphene FET biosensor to detect BNP. BNP actually is a disease biomarker related to heart failure. Similar to the other to the previous work, actually this is graphene FET. To increase the sensitivity, we decorate a golden nanoparticle on the graphene sheet, and we immobilize the antibody on the golden nanoparticle surface. Because the BNP is specific to the antibody, so the BNP from the serum, okay, so can be detected. And this actually is a microfilter system, okay. So this means the serum can filter by this microfilter system. By combines the biosensor and the field system, okay, the micro system is developed. We can use this micro system to detect BMP from the blood successfully. And we can do real-time measurement. And you can see we add different concentration of the BMP. And the current decrease. And by this work, we define limit of detection of this device is 100 femtomolar. Very recently, okay, we work with Peking University to develop a Wafer-Scale uniform CNT FET biosensor, which can be used for detecting the disease biomarker. Actually, this is the schematic diagram of this work. Actually, this is a wafer, and CNT is deposited on the silicon oxide surface, and then source drain is fabricated. And on the CNT surface, yttrium oxide was also deposited, and on this surface, we decorated the golden nanoparticle. And two example was presented. We actually detected two biomarkers in this work. One is DNA, and the other one is tumor derived microvesicle. To detect the DNA, it's obviously we can immobilize the probe DNA on the surface. And to detect the microvesicle, we actually use aptamer because aptamer is very specific to vesicle. And this work we can detect DNA as low as 60 attomolar. And for MV, we can detect 6 particles per milliliter. So what is the Ebola virus? Actually, similar to coronavirus. Ebola virus is a single-stranded RNA virus, and is severe, often and fatal. So current method similar to coronavirus is based on RT-PCR and antibody detection. So question is, can biosensors directly detect the Ebola virus particle? So the answer is yes. So we use this biosensor for immunodetection of Ebola virus. So the principle is quite simple. This is a graphene-based FET device. And to detect the Ebola virus, we only immobilize specific antibody on the RGO film. Then the Ebola virus can be detected. So this assay makes direct detection Ebola virus particle possible. So several characterization were done before detection. Actually, this is a TEM image. Okay, the Ebola virus you can see very clearly. Actually, it's a filamentous virus. So about 900 nanometer in length and about 17 nanometer in diameter. And this is the fluorescence experiment. Okay, the reason is okay. We want to verify how antibody is mobilized. Okay, so after antibody is mobilized, we use a fluorescence dye labeled antigen. Okay, then on the microscope, okay, the red signal means antibody has been anchored on the surface. And the efficacy is a SEM image. You can see actually this is 2 electrodes, and in between we can clearly see this Ebola virus. Okay, this means the Ebola virus can be captured on the chip surface, and that potential result shows this virus is negatively charged. So to verify the stepwise of functional process, and we use the electrical characterization of this FET device. Actually, this ambient polar curve, as I shown previously, and RGO, this black curve is represent RGO, and we use the PAS. Actually is a link molecule, okay, because this molecule contributes to the p-doping, okay, so the direct point shift right, and after immobilized with antibody actually is negative charge, so the direct point shift to the left, and after Ebola virus is bound to the antibody, because as I mentioned earlier, the Ebola virus is negatively charged, so the direct point shift left again. So this means the functionalizes process is successful, and stability and reproducibility work also done. For example, in this work, we choose 24 device, and the IV curve you can see the IV curve looks very similar among 24 device, and the direct voltage is centered at 0.425 voltage, and this is actually stability experiment. We store the chip for 15 days, and over 50 days we can see the signal doesn't decrease very much. Now, this means this device has good reproducibility and stability. Sensitivity, we did this work very similar to the DNA work. Okay, we applied different concentration of Ebola virus. You can see the direct point shift to the left, and by this experiment, we found okay LOD as 2.4 pico per milliliter. Specificity, we perform actually 2 experiments. One is okay, we change okay target okay for in this work okay we choose H1N1, H3N2, H9N2, replacing Ebola virus. So you can see the signal from the non-specific targets is quite small compared to the signal of Ebola virus. Similarly, we choose different protein replacing the specific antibody, like EPCAM, CD9, and strep RBD. Similarly to the results, the signal is quite small compared to the signal of specific antibody. And we also did the reversibility experiment. So in high ionic solution, okay, so actually Ebola virus and antibody can be dissociated, and we do 3 cycles of association and deassociation, and you can see the voltage, and from the first one is 120, and the third one is 19. This means the reversibility is good, and we can say this chip can be reversible. The last experiment we did is to use this biosensor to detect the Ebola virus in blood sample. So we dilute okay the blood the serum with different concentration, 10%, 30%, 50%, 70% and 100%. And we spike, okay, different Ebola virus to this serum. And from this figure, you can see, so if the serum is diluted, the signal is quite high, because in diluted serum, the interference is small. And we can see, even in the 100% serum, the signal also is very high. So this means the biosensor is capable of detecting Ebola virus in blood sample. So last part, I would like to share with you ongoing
Professor Zhang, how many slides do you still have?
Sorry?
How many slides do you still have?
Only maybe 5 or to 6. How many minutes I have?
So how about to give you another less than 5 minutes? We need to move quick. Thanks.
Oh, sorry. Okay, I move quick. So because of the virus, okay, our laboratory was reopened last month, and my student can work now. So we are targeting actually 4 works. One is actually we are going to use RGO-based FET biosensor for PCR-free detection of coronavirus RNA fragment. Okay, this is actually different from the RT-PCR work. So based on the first work, we are going to use this biosensor array for multiplex detection of coronavirus RNA. Okay, so in this case, we are going to detect 3 specific RNA fragment from the coronavirus. The third work, okay, different from the nucleic acid detection, we are going to use the CNT FET biosensor for lab-free detection of the actually N protein. And the fourth work, based on the previous work, we are going to use this sensor to simultaneously detect multi-protein. And I will go quickly. Okay, to summarize my work, actually, we are going to miniaturize, okay, our device and do integration. And finally, we will do a multi-functionalization. Hopefully, we can have nucleic acid detection region here, and the protein detection region, and control region. And we can combine carbon material-based FET array biosensor with microfluidic system and ASIC readout. Okay, hopefully we can fabricate a portable device. This device can be used for rapid and accurate detection of the COVID-19. So lastly, I would like to thank support from the National Science Foundation of China. Also, I like to thank my collaborator, Professor Zhang from Peking University, and Wei Hua and Zilin from Wuhan University. So also, I like to thank member of my team. Lastly, thank you very much for your kind attention. Thank you.
Thank you, Professor Zhang. Definitely, there are lots of work. You have lots of good work you have been doing in this area. That's also why we got multiple questions to you because of time. So how about we choose one? So the question is, how electrical conductivity affect the detection of DNA?
Sorry, I can't hear you very clearly. You mean the conductivity, right?
Yeah, the conductivity of this, I think the material used as a sensor affect the detection of DNA.
Okay.
Yeah.
Yeah. It's a very nice question. Actually, okay. So the conductivity is very important. Okay. The reason we choose graphene or carbon nanotube is because of the good performance of conductivity of this material. So actually, because of the DNA is negative charge, and the signal can be high enough. Okay, I show you one slide. Okay, one slide, and yeah, you can see the change of Dirac point. Yeah, from minus 50 to minus 200. So this means we think here the signal is good enough for the detection of virus.
Okay,